Traceability. Assalamualaikum warahmatullahi wabarakatuh. May peace be upon us all. Om Swastiastu, greeting of virtue, Namo Buddhaya. Good morning, good afternoon, and also good night for the speakers, moderators, committee, and also both international and national student participants. We would like to extend our warm welcome to all participants of the fourth day of Short Course 2026, Nature, Science Tech Innovation, Co-Creating Sustainable Agri-Food Industry, and Climate Solution for a Resilient Planet, which is organized by the Faculty of Agricultural Technology, UGM. My name is Indah Kusuma, and you can call me Puput, and I will be the MC for this morning session. And today we are going to have our some session with Associate Professor Dr. Weijie from Nanjing Agricultural University, China, already attending in this room. And the second session with Associate Professor Dr. Santat, which in short from Prince Songkla University, Thailand, that will joining us also in this room. And the third session with Professor Lili Sutiarso from UGM will also attend in this on-site participant. And Dr. Melania Casertano from Wageningen University, Netherlands, that also will attend by online in Zoom meeting. They will share their knowledge and experience to expand your understanding. And also its discussion session will be led by a great moderator that knows of each expertise. Moving to the first session, the topic is about from Spectra to decision AI-driven chemometrics for fast classification, prediction, and traceability by Associate Professor Dr. Weijie from Nanjing Agricultural University, China. Mr. Weijie is standing on site in this room. 早上好，老师。Not yet. And the session also will be led by our moderator, Dr. Lulum Liliana. And first, let me introduce our moderator for the session by reading the moderator CV for all of you. Dr. Lulum Liliana is a lecturer from Department of Food and Agricultural Product Technology, UGM. Her research focuses on phenolic compound extraction and antioxidant activity in food metrics. And she has published multiple papers in international journals, such as Trends in Science and International Journal of Gastronomy and Food Science. Her recent works are about encapsulated plant extract as potential clean label alternative to synthetic preservatives, potential of graphene oxide as multifunctional platform for active food packaging, and Naniora traditional Batak fermented fish. Without further ado, please welcome our moderator in this session, Dr. Lulum Liliana. The time is yours.
Okay, thank you for the MC. Okay, and good morning, everyone. A little bit tired because you already have a field trip, a full day field trip. So welcome to the fourth day of summer course. My name is Lulum Liliana, and I will be your moderator for today's session. And before we begin, since we are very fortunate to have our speaker on site, let's start with a quick interaction. How many of you familiar with AI, artificial intelligence? Yeah, I think everyone using AI, even for like just simple things like let's ask ChatGPT, let's ask Gemini or anything else. So yeah, today's session we will talk about artificial intelligence using of AI in the food sector. So, and that was a really great session. And it is my pleasure to introduce Associate Professor Dr. Weijie from Nanjing University, from Nanjing Agricultural University, China. And he completed his PhD through a joint program with INRAE and Avignon University, France. And his research focuses on artificial intelligence in food engineering and infrared spectroscopy. particularly on rapid and non-destructive food quality and safety analysis. He has published in a leading journal such as Trends in Food Science and Technology, Comprehensive Reviews in Food Science and Food Safety, and Food Control. Before we begin the session, since Dr. Lan here with us on site, I would like to ask a quick question. How was your experience been so far in Indonesia? Is this your first time visiting Indonesia?
Yes, this is my first
Okay.
time, and it's a very nice experiences
Oh,
into this very magic country, actually.
yes. Yes. We hope you enjoy your
Yeah.
time in
Thank you.
Indonesia. And before we begin, allow me to briefly explain the session flow. Dr. Lan will deliver his presentation for like 75 or 80 minutes, and also followed by 20 minutes Q&A session. And without further ado, Dr. Lan, please, the floor is yours.
Hello. Okay. Thank you and good morning. It's my great honor to. This is my first time to Indonesia, and I'm very happy to share some our research and some our outcomes relating to spectroscopy and imaging techniques coupled with AI power and for the agricultural materials and the foods and nutrition and safety. So I made some small modifications for my research title. So from Spectra to food decisions, AI-driven chemometrics for fast quality prediction and smart processing control. Actually, we want use these kinds of advanced technologies not only on the quality of the raw agricultural materials, but we want to control or develop the smart food processing control as well. So I add this part, and I will introduce some of the research outcomes of that. So first, I would like to thanks for our contributors for this work. I'm not alone. So we have a big research team, and we have several collaborators, including some researchers from French, from France in Haye, and also from the Belgium and Wallon Agricultural Research Center, and also our Chinese team. So we are also supported by several research fundings. So first, please let me have a very brief introduction. So I'm from China, and I'm from Nanjing Agricultural University. So I finished my PhD at French National Research Institute for Agricultural, Food, and Environment in France for 4 years. And after that, I back to my country and to have a job at Nanjing Agricultural Research University, Agricultural University, College of Food Science and Technology. So till now, I'm in charge of the Faculty of Food Science, Food Security and Safety. Okay, I will share these outcomes in 5 parts. First, I will have a brief introduction for Nanjing Agricultural University. So this is our new campus, and this new campus is we started this a very big campus, and we start this new campus in last 2 years. So actually, we have 5 different campuses in Nanjing City. So this is the biggest one and the newest one. Sorry, it's a little bit slow. Okay, so here it's okay, here it's the Nanjing City marked with yellow color, and it's very convenient. It's at the it's at the southeast of China, very close to Shanghai. Take high-speed train with 1 hour, and we take to the Peking and in 4 hours. So it's a very convenient and very historical city. So in our agricultural university, and we have 5 facilities. So the first one is the facility of plant science. And the second is the faculty of animal science and biology and environment, and also the food science and engineering. What we are research faculties is at the top right part is the faculty of food science and engineering, and also have the faculty of social science. So I will have a short, more detailed introduction of our university tomorrow. So we have 14 disciplines are ranked in ESI for top 10,000 for in 2 disciplines is plant science and animal science, and also the agricultural science. And we ranked the top 6 the best global universities of agricultural science. And also, we have over 100 national and ministerial provincial levels of research laboratories centers. So we have the National Key Laboratory of Group Genetics and germplasm enhancement and utilizations, in particular with rice, wheat, cotton, soybeans, vegetables, and pears. So we have the very highest, very famous, top research centers in China with relating to this. And also we have the only one top state key laboratory of meat quality control and culture meat development. So culture meat is a future food tapes or the food tapes. That's it. That we have the first culture meat, developed culture meat in China. And second is our College of Food Science and Technology. So in our college, and we have over 100 staffs, so 120 faculty members, so 32 professors, and almost all of the members have doctoral degrees with 60% of overseas researches. And we have 5 research directions, including the food nutrition, food microbiology, and biotechnology, meat processing and quality control, and processing and quality control of agricultural products, and Glock mix and Glock bioengineering. So what our team is located at the processing and quality control of agricultural products. So this is our team. We are called NDT team. It's non-destructive testing research team. So we can also say the NDT lab. And in our lab, we have 4 researchers, Professor Kang Tu and Professor Lei Qingpan and me, and assistant professor with Yang Chuanyu. And now we have a team over 60 students, including 20 PhD students and over 40 master students. Okay, this is just a short background. And so let's back to our research background and spectroscopy technique. So let's first think about what is the main challenges, one of the main challenges in agricultural and food system. Because nowadays, and we really call it sustainable food system or sustainable agricultural system. But if you want to reach this objective, you should find a very balanced point, because you should meet the society's demands or customers' demands of food safety and quality first. And also, you should keep the sustainable food products and incomes. And also, while you produce or while you grow the food or agricultural products, we want lower energies and waste to make full use of these materials. So we have to be made satisfied with societies, producers, and environment. But, however, now, and we have lots of variable factors that can give the returns or give the unstable to the agricultural food system. First is the genetics, because we have lots of different varieties, lots of different cultivars. This is the first. And then we have the growing conditions. So maybe the irrigations, orchard management, and so on. And also we have the processing conditions and different variable, durable processing strategies, and to make lots of different kinds of food. So variability of raw materials and processed products is the main challenges. And this is can be written or it's a major task deal with for the sustainable agri-food system. So we are thinking about how to facilitate and optimize choices to produce sustainable food products. We have lots of researchers now, or lots of students study with the horticultural research. We're mainly on their crop load, irrigation, years, harvest, date of harvest, and so on, with their agricultural qualities. And also we have some food scientists. Now they are working on the different processing technologies on the food safety and quality. But we want to find some solutions to combine the raw materials and their processed products. We need some technique and to combine this whole supply chains with the dealing from raw to the processed food. So this brings out the main concerns. So limited knowledge about between the raw and the different processed kinds of food. And why we want to develop efficient and smart strategies to identify and manage this variability or variation for food raw materials and their processed products. So when we want to control these procedures. Spectroscopy, maybe some students or maybe some colleagues are familiar with, but some of them maybe will be far away. But we want to say spectroscopy is an ideal analytical tool for industry. Why? Because it's rapid and can be non-destructive or non-inverse, and also it's high throughput information. For example, here we give some informations, give some different technologies such as infrared spectroscopy, and this is a non-destructive one, near infrared, with rapid and the advantages of rapid and non-destructive. And also we can use mid-infrared, and mid-infrared spectroscopy can give the fundamental vibrations and will define the bands for organic function groups. I know some students focus on on some materials development or some functional food or bioactive composition. So you can use the mid-infrared spectroscopy to identify some bioactive compositions. And you can use near-infrared with non-destructive evaluations with raw food materials and also as well as the processed food. And we can combine not only the spectroscopy, but also the high-dimensional information. For example, imaging information from 2-dimensional to 3-dimensional. And so this is we can combine the images and spectroscopy. We call it hyperspectral images. So we have not only the spectroscopic information, but also their spatial information. So we give the distribution of the chemical compositions of the materials, of the food materials. As well as we can have the more sensitive information images. For example, the Raman images, it can give us scattering lights of some very very limited compositions, and it's with sharp peaks that near infrared. So we have these powerful and advanced technologies, but another very important thing is that these kinds of informations can be applied both with at orchard, at harvest, and food storage, and during the process. So all of these supply chains we can cover use the same or use this kinds of technique. So that's why we can combine all these informations together to give the combination of the high throughput data and to analyze them, and in order to control the whole food supply food chains. So that's why we developed efficient spectroscopic methods and new resolutions to manage the raw materials and the final product's quality. Okay, here is our colleagues and also one of my supervisors from Wallon Research Agricultural Center at Belgium. And we are almost the same, paralleled, because last slide, and we share some very cumbersome instruments. But actually, with the development, with the technologies, we can have a very small and very small and very convenient sensors. You see here, at 2000, that means 25 years ago, and lots of researchers use very cumbersome techniques, imaging and spectroscopic techniques. But now, after 10 years, there are more and more portables and microsensors for the food industry and for the agriculture. And now they have a multi conditions applications, such as the apartment mirrors, baby nears, near images, near benchtops, online nears, and microscope nears, and so on. And so from the scientific research to the industry applications, so they will have lots of different kinds of equipment for the food science and technologies, and also for the agriculture producing. So not only on the images and the spectroscopy, and one of our main subjects or one of our main concern is that we want to understand what of the light interaction with the food tissues. So here we do some fundamental research work, not only on technology work, and we want to know the light photonics and how to interact with the food tissues. So we know once a shade of light penetrates on a sample, it can have 3 status of the light. First is the surface reflection, and also the scatterings. That means they penetrate inside of these samples, and some of them, and with photons, will be absorbed, and so it's an absorption. But some of them will be scattered and penetrate through or reflect. So they have 3 different types. And now we can use some single spheres systems, optical systems, to characterize only this informations, photonics. Why we take these informations? Because if this absorptions is mainly to the chemical compositions related to, and for the scatterings, we are related to some structure. That means internal structure information. So if we can extract absorption and scatterings information, and we can evaluate individually or more precisely with their chemicals or the textures information. So that's why we have the fundamental study of interaction of light with food tissues. So here is NDT Labs equipment. First, we have electric sensors like electric nodes, visible, near, mid, infrared spectroscopy, and Raman spectroscopy. And we also have advanced image techniques in our lab, machine vision, hyperspectra images, and hyperspectra micro images. And also we have the optical properties to understanding the fundamental light interactions with the food tissues, with single integrating sphere systems, and the special frequency domain imaging systems. And finally, we have the self-developed equipment with portable rapid detection devices and online quality detection platforms. I will introduce some of our works with that. But previously, I will introduce the not only based on techniques, and what we are also focused on the chemometrics. So we use some algorithms, and we use some new methods, deep learning, machine learning, and so on, and to give the evaluations of raw materials with linear and nonlinear regressions with their qualities and their spectroscopic information. And also, during the processing, and we have the raw materials and their final products. So we want to have this kind of information and final products information and to analyze their correlations during the processing, while the different processing strategies, to know what's the structure and chemicals modifications during the heating or pressure or grinding or refining or this kinds of processing strategy, this status. Okay, and finally, we develop some new chemometric methods to design new products by artificial intelligence. So use these kinds of high throughput information to design the new products let computer think by themselves and to give us the strategies of new products. So I will introduce our outcomes of that. So let's move to the research outcomes. So first, we used the electric nose. This is our self-developed one systems with mini electric nose. So we use this kind of equipment, and we evaluate the different stages of strawberry and cabbage, as well as we evaluated the different drying stages of the mushrooms. And so we have this kind of information to let the orchard producers and the industry manufacturers to know that which is the best stages of drying and their freshness of the strawberry fruits and vegetables. As well as we use the near-infrared spectroscopy to non-destructively determine their maturing stages of grape. So we can acquire such a spectra and this kind of, and we use some of the signals related to their color and also related to the internal sugars and acid content. And finally, we can develop some models to classify their different maturing stages and also to give predictions non-destructively with their sugars and acids, and to give this give the quality and safety in assessment of this grape fruit. And further, we have researches on the microscopic hyperspectral images. So in these spectral images, so we have this kind of techniques, and this is we can have the images with spectroscopy at each pixel of spectra. So in each images, we have over 10,000 of spectrum. That's with also first image with one of the images, and we took these images with the cell shapes and cell structures, and we extract with the cells shape and with the cell walls and with the intercell spaces, and we extract this information for the for the analysis with their spectral signal. And related to their pectins, hemicellulose, and cellulose degradations during the apple's firmness softening. And well as we have the post-harvest disease of the pears, we have the several surface of the damaged surface with the damaged surface stages. And we collected these stages. You see here, we have the very, very clear degradations clearly with this cell walls weakens of the images after we treat it. And in fact, this cannot be visualized by our traditional conventional images. And it's very, very hard to study with your traditional chemical analysis, such as GC, HPLC, and so on, analytical methods. Okay, after that, and we have some fundamental research with optical properties. So as I said, we use the optical absorption and optical scattering. Optical absorption related to some chemical compositions because it's the photon absorbed by some waters, sugars, acids, polyphenols, and so on. So this bioactive, these informations, we develop some models with using the predictions of their chemical compositions, such as for example, such as their sugars, maybe on polyphenols, total polyphenols, and so on. So we can use that to directly predict their polyphenols, sugars, acid, and so on. And in terms of their scattering property, as I said, scattering property is related to the, sorry, okay. As I said, the scattering property is related to the structure. So in generally, when we want to detect the structures of cells, and we need to take images, and we use some software to calculate their surface and to calculate the numbers of the cells, right? And you use the microscopic images. But for us, we use the optical properties. We can directly predict their firmness, their cell walls, their cells diameters, and their cell wall thickness, and also their permeability of the tissues. It's very rapid, and it's quick, and it's more convenient. So this is the advantages of these techniques. And besides of the fruits and vegetables, and we're using the machine version images with the industry to online sorting the damage of the eggs. So we have the images of different colors of the shells, eggs, and also these eggs have some cracks on the surface. And generally, in the industry, we will, with the machine version, we will have a very, very fast sorting per second, over hundreds or thousands to thousands per second with the online sorting. And in this technique, and we can have the different processing images methods to extract the cracks features and with on the images, and very rapidly. And finally, we give the decisions of the damage, the levels of these eggs based on image techniques. Also, we have the hyperspectral images. That means hyperspectral images has the advances compared to the machine version because it can give some way that we cannot see the internal damages of fruits and vegetables and food. For example, here we have some damages under the surface. This is the peach fruit, and we have some damages under the surface or internal damages of fruit. And we can use the hyperspectral images to give some spectral informations, and we find some features, informations to visualize their internals or undersurface damages. And we use the rolling stars with the images to have the global surface of this fruit and to see their undersurface damages with the online sortings. And also, for eggs, we use the hyperspectral images not only on their damages, such as cracks, and we can also use them to predict their internals or surface safety and quality. For here, this is a visualization of the egg samples and different levels of contaminations. And we can visualize their surface of the contaminations levels. And also, we developed some models with eggs quality indicators like yolk index, pH, protein content, internal protein contents, TVBNs, and total bacterial contents, and so on. And so we can use this one technique to give the assessment of lots of quality indicators. Here is our self-developed by our student, master student. And this is equipment with near-infrared spectroscopy. And as we know, we have some maturation stages, judgment with this, with graphs. So our students developed part ball devices, very small, and to detect non-destructively put the graphs here. And we can use our own designed software to give the informations of their maturation stages and sugar contents and acid content and directly. And this is our, what we mentioned is our mini sensors, electric nose sensors. So this is the air pump switches and the air rooms. So we have the air chambers, and finally we have the micronear pumps. We find some channels related to design this with, we find some channels related to the freshness of the fruit, and to make this very low cost and very fast with the development of this fresh with fruit freshness evaluation. And also we use this key sensor units, and with mobile of the strawberry from this point, to evaluate all of the strawberries freshness, and with all the evaluations of the strawberries in freshness. So it can tell us with a large number of strawberries with their qualities and safety. And also this is our eggs online sortings by our labs, designed by our labs. And we can have the yolks, because I don't know here, and because in China, sometimes we have double yolk, the eggs. But some of the egg we need, but because Chinese people prefer double yolks eggs, some of them. But we want to use non-destructively techniques to select them. So here we use the machine version, design a machine version equipment, and software to detect, very rapidly detect the double yolk eggs to select these kinds of products. Oops. Sorry, it's a little bit. Okay. And this is also our developed the partibles detectors for the drying stages. So we put some drying stages. We developed the images, multispectral images detectors, and we put the drying fruit slides in this machine, and it can be rapidly evaluate their drying stages or qualities, such as some bioactive compositions, chlorophylls, and also the water contents, or with all of these surface or all of these samples distributions, and give their judgment of classification of their drying stages and nutritions. So for the org chart, and we can use the near sensors to classify the different stages, maturation stages. This is what we work in France, and we have a different org chart, and we use the fruit thinning and non-fruit thinning works, and we can have the fruits growing to be the best to design to make decision for the best optimal harvesting date to evaluate their sugars and tetraploid contents and dry matters and silver content. And what we are more concerning is the correlations between the fruits and the processed. So firstly, we characterize the quality of apples, lots of texture chemicals and biochemicals, and also as well as their processed purees. So we use these kinds of techniques. We use these parameters to find the correlations with these quality parameters. And we find some interesting things that with the firmness of the fresh fruits is really correlated to their processed purees rheology properties, as well as dry matters, sugar contents. It's related to the stable during the thermal processing. So based on that, and we use the infrared spectroscopy to non-destructively with the nears and to predict their qualities of purees. So we can use the non-destructively near information to predict the final quality, the processed quality, to give the guidance of their processors of your what's the final qualities of your processed product of your process based on your raw materials. So in order to this, and we have the institute near detection devices for the purees. So we have this self-developed the institute optical sensors, near sensors, that's integrating also the temperatures, pressure, and grounding sensors, and to acquire their chemicals, physicals changes of in these conditions, in different conditions. And we collect lots of data and to find the relationships between this multi-field coupled data and in order to control their qualities. And in this way, and we can control the qualities during their processing. Here is a near sensor, and we put at the fish homogenates in this way, and we can dynamic control their processing conditions with the temperatures, with the grinding and the refinings, and we can have the changes, we can have this control of their qualities to give their sugars, organic polyphenols, predictions, online predictions, and dynamic control. So we are thinking about the food products, and we are talking about some previous outcomes of the raw materials. We are also talking about during the processings. And what we are finally, we want to develop the new products. Actually, for example, if you have the different kinds of single composed products, for example, you have different varieties, you have different kinds of juice, you have different kinds of cereals, you have different kinds of oils. So think about if you want to develop the personalized products or new products with the mixture of these kinds of food, how will you do? But generally, you will based on your experiment, and also you will based on maybe your knowledge about how to balance the nutritions and balance the taste and balance the colors to find the best percentage of the mixtures and formulation strategies. But here, we want to build the intelligence food formulation systems based on infrared to guide the new and personalized products for different kinds of peoples. So what we will do is firstly, we do an experiment with different apples processed to the puree. You see, there was a very large different changes with the appearances and also their rheological properties and also their internal nutritions. And then we doing the mixtures of different percentage of that. But we also acquired non-destructively with the infrared spectroscopy. Afterwards, we developed a new chemometric methods that we can make the fusion with based on curve resolution, alternatively squares. And this is we can extract their concentrate profiles to reconstruct the spectra. And this method is developed by us. And if you are interested, you can read our papers. And this is what we want to propose in the future with all the different kinds of food for the formulations. And here, what we can do based on that, and finally, we can use only the spectra of these 4 non-destructively apples. So we use the non-destructive apples spectra, and we developed using our chemometrics methods, and we input this data to our models. And finally, they will provide you with over 100 different kinds of formulation strategies, and they also as well as their processed qualities, such as the different taste. What will be with these kinds of formulations with the rheological properties, colors, and so on. So it can use this. And also this technique, I designed with the paper's cover page. And this is also the first paper cover page of this journal. And we are proposing the smart food formulations based on infrared spectroscopy. And all in all, we want to propose the smart food processing, a new topic with smart food processing based on AI and the spectroscopic techniques. And we want to do first is the raw materials control. That means we want to sorting the damaged raw fruits or raw agricultural products firstly. And then we want to find the suitable products for the processing. Maybe these fruits will have higher water contents and it will be suitable for juice. But for the lower water contents, maybe it's for drying and so on. And this is more intelligent and suitable. And then we will use the spectroscopy to give the online detections or online control, precise control of processing, to online control their qualities, dynamic changes, and modify automatically with their heating times, temperature, pressure, grinding speed to produce the expected food and stable quality during the processing. And finally, we want to use the chemometrics, such as our self-developed chemometrics, and also the artificial intelligence methods, and combine the consumption data, marketing data, and let's put the systems with infrared spectroscopy to produce the new products automatically and to give the market. And this is, we called it, the whole of that, we call it smart food processing. Okay, from raw materials and during processing until to the final personalized product. Okay, so that's almost all my research introduction. And here we want to introduce a journal that with food innovation and advances. And this is a journal that with open access, and it's a ESI journal with, it's a very good. And the second, we also like to recommend another journal is for the Journal of Future Foods. And this is also with a very high impact factor over 10 at this year, 2026, and with ranked with the GCR Q1. And it's also a very good journals. And I'm also one of the editors of these two of the journals. So we are recommended all of the students, if you are interested, you can submit your works and to our journal. And specific thanks to our colleagues and to the foundations and to our joint collaboration universities. And we are also seeking some collaborations with Indonesia. And so thank you. That's all my presentation. Thank you.
Okay, thank you, Dr. Lan. That was a very insightful and comprehensive presentation. So we have, now we will not have a Q&A session. So for the on-site participants, please, you may raise your hands. Yes, for to ask the question to the speaker. And for the online participants, you can write the question in the chat box. So please, for the first session is three questions.
Hello. Good morning, doctor. I'm Kenneth from the Philippines, in Delaseu. So, first of all, I'd like to, listening to your discussion, I was intrigued with your technology of detecting good produce and bad produce. I'd like to ask if there would be any cheap alternative products from China that uses this technology, and that local farmers in Southeast Asia countries can use locally.
You means if there are any
technology or products that uses your AI that us Southeast Asian countries can use
Okay.
that is cheap, so that our local farmers can use.
Okay. Yes, that is what our objectives. So we want to develop the very cheap portables and very convenient sensors for maybe for the orchard and for some industry producers, because for the agricultural producers, they cannot suffer from very high the price of the equipment. So now we have some portables sensors with fruits and with apples with lots of orchard to use. And we developed the models. We developed the prediction models for them. So in general, it's sold in China, and we have lots of industry collaborators for spectrometers, and they use our models and to divide and to give their products, spectrometer products for the orchard. And we sell about at least at least I think 800 spectrometers per year for in China orchard with the for assessment or the best harvesting date and the qualities of apples, pears, peaches, and grapes and strawberries and so on. Yes, and also we have collaborations with the online sorting, the industry online sorting, and this is a very big country. Big company, and so it have lots of machine for Asia country, European country, and so on. So we developed some models, online rapid sorting models for them with the surface damage and the internal quality determinations of fruits. Yes, so now the near sensors or the portable sensors is relatively cheap. The price can be reached around, I think the prices reach around, I think $200 to $500. That's it. So, but it's multifunctional. So you can use with lots of different kinds of fruit and lots of different kinds of fruit. That's it. Yeah.
Thank you so much, Doctor.
And another explanation, and we have some collaborations with the refrigerator, and we use the image sensors. Want to use the image sensors to dynamic evaluate their freshness during the cold storage of your refrigerators. And now they have the products. And this camera, it's only I think only $20 to $30, and with these cameras, and with the refrigerators for the freshness characterization evaluations of your cold storage products. Yeah. Okay.
Okay. Yeah.
Okay. Thank you.
Good morning, Doctor. My name is Mark from the Philippines. You just mentioned regarding the freshness and using this AI technology, spectroscopy in detecting the freshness. No, I just want to ask in regards to this technology. I want to know what are the challenges, especially when it comes to global food waste, and when it comes to large-scale industrial applications. What are its limitations?
Okay, you want to ask the limitations, right?
Yes.
Yes, this is a very good question. And the limitations for spectroscopic and for the imagings, hyperspectral imagings, I think there are two. The one is the price, because the price, if you want to acquire the very accurate predictions, and you have to use the more informative signals. But if you want to use more informative signals, you will increase the price of the equipment. So you should find that means that's what's our objective, because you should find the balance. That means, for example, for Philippines, we know there was lots of bananas for banana fruits. Actually, for banana, one of the main qualities problems in China is their cold injury. That means the storage of the cold injury, different temperatures during the transportations, and But we want to detect this. We want to find the cold injuries related the very special signals. So if we find these special signals, we will very efficiently reduce the cost of the equipment. So this is the challenges. So we need to find the relation features, spectral features. So that's because in generally, if a high output information spectrocalorimeter, it will it will have around 400 channels, different spectral channels. So they require the information by channels. So it will be very slow. But if you want to if you know which channel related to cold injury of banana, so you can select this. Maybe you can just select 10 channels or 20 channels. So in this channel, you will decrease the cost, because other channel you will not use that, and also you will increase your speed of detection. So this is the limitations of that. Another is that we have the data. We need to have the acquired lots of data, and this is very valuable. And this is a key. That means a core support of your technology. That means someone can copy your equipment, but for the models, for the database, and this is yours. So this is very valuable, and for that. So yeah. Okay.
Thank you, Doctor.
Hey.
Okay, yeah.
Right.
Next.
Good day, Dr. Weijie. And I am Sam from Philippines, and I see a lots of opportunity with NIR spectroscopy. And one thing I'd like to ask is it possible that we manipulate the food quality using spectroscopy or integrating technology with spectroscopy to reduce food loss or food waste?
Your question is it possible for use infrared spectroscopy to reduce the food loss?
Yes, to manipulate the food quality, like
Manipulate the food quality.
yeah, manipulate. Yeah.
That is that means we in generally we use the infrared spectroscopy to predict to give the assessment of the food quality in terms of not only the freshness, these very integrated indicators, but also their very detailed qualities, as well as talking about maybe the food waste. And sometimes you during the post-harvest, for example, and such as strawberry or some berry fruit, if they have a very small damage with mechanical damage or some bioactive infections, they will spread very fast with different fruits, and so we need to detect them as early as possible. So that's it. Why we use this kind of technique to reduce the loss of during the post harvest. You understand what it means? Yes, that's one thing. Another thing is that we also, for the fruit waste, I think we need to combine the different technologies, not only on spectroscopy, because spectroscopy is for the detection, mainly for detection and quality control. But as also we use some, we also need to combine some other technologies for the food waste valorizations. Yes, that's it. Thank you.
Thank you, Dr. Weijie.
Yes, okay.
Good morning, doctor. I'm Hannah from Vietnam.
Okay.
First of all, thank you very much for a very interesting presentation. And I also excited when you mentioned about double yolk egg, because Vietnamese people love double yolk egg too. So now relating to your research, could you tell me how about samples that you use in your research for detecting the crack of eggs?
Yeah.
And how many percentage of those samples used for training and testing?
Okay, so it's a very specific question. Yes, because now in the lab scale, and generally we will use around hundreds of eggs, such as in the cracks, we used around 500 eggs. And this is the first for calibration, for models calibration. And for the validation, and we will use a set of around 100 to 200 samples. But this is only in the laboratory. And then we will take these models. Now, if we have a project, we will take these models to the industry. So it will have thousands or more and more to optimize our models, to make it more accurate. Yeah, to validate these models. Yeah.
So like the percentage of like training sample and testing sample, like
3.1 ratios.
Okay. So that's what the percentage is based on, for your opinion, or like somewhere?
What's
the percentage of testing and training
okay?
sample?
So in generally, it's the sample. You means the mark with crack or non-crack?
Like you mentioned about like one third, right? for calibration and validation. I mean the percentage of sample for this step.
Percentage of the samples?
Yeah, it's what were they based on?
Okay.
For your opinion or for like
yeah, for industry. For industry manufacturing, yes, for industry. They will give us the definition of the cracks, the levels of cracks, because the different cracks have the different levels.
Yep.
And generally, they will visualize, they will by their eyes to judgment. And we will find them, sample them. That means marked with the different levels of the cracks. And then we, based on their experiences, they take these eggs to do our models.
Okay, thank you very much.
Okay. Ed?
Good morning,
morning.
and welcome Dr. Weijie to Indonesia. My name is Eda Milamsyah from University Putra, Malaysia. I have concern regarding lab-grown meat. Lab-grown meat. You see, as far as I know right now, majorities of the scholars and Muslim countries and Muslim majority country consider lab-grown meat as a gray zone. Basically, it's not halal because it was not slaughtered in Islamic ways, because we don't know the origins of the meat itself at the first place. So my interest is basically, is it possible at the future we had a seafood lab-grown meat instead, like tuna or salmon lab-grown meat? And I believe that if it's possible, we can branch out to lots of opportunities, especially the Arab country, you know, the sheikhs.
Yeah.
So yeah.
Yeah, this is a good question. And even this is not related to spectroscopy, but this is for the culture meat. You means culture meat, right?
I would like to know on the unknowns.
What is
basically the origins?
Origins of
Lab-grown meats.
Origins of the meat. Now we do limited works with this origin of the meat. So we more focus on the quality of the meat. That means we focus on the safety and the quality of the meat products, raw meat. We didn't have experiences with the identify the different regions. You mean that? The different regions of the meat?
The content.
The content?
Yeah.
Oh yes, for content it's okay, because we evaluate their proteins, their lots of, actually I don't know how to express it in English actually. Sorry for my poor expressions, but we have that. We have rapid determinations of this, such as beef and beef proteins and safety and proteins and so on. Lots of, and fat and so on. That's a fish, beef, and chicken meat, pork meat. Yeah, that's all. All of that we can. It means infrared spectroscopy we can have the ability to evaluate that. Yes, but we didn't present that. Actually, we have lots of experiences with a lot of different kinds of food. We have over 2 decades of experiences with working of that. But here we didn't present the meat, concerning the meat. But if you're interested, you can search my name, and you can have lots of publications concerning with the meat. Lots of, and you can find some technologies maybe you're interested with the qualities, with and also the some maybe your traceability, but traceability we didn't work that. Okay, thank you.
Okay, thank you. So guys, we still have more time, so don't be worried. If you still have questions for the speaker, we still have like 30 to 35 minutes. So before that, I would like to read the question from the online participants. In predicting the freshness or quality of fruits, for example, the sweetness of an apple using chemometrics, what is the common sample size to build some sort of robust models? Yes.
So that means you means the for source processings, the
Yes.
models?
Yes. What is the common sample size to build some like robust models?
What put model?
Robustness.
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 So it depends because for the model development, and we will concern is actually most of the researchers or most of the new beginners with these modelings, they will concerning about the models with lots of sample numbers. But this is not right, in my opinion, because what we want to develop the models, we will to think about two parts. The first is the variability of your samples. That means, for example, if I want to take general models for prediction, I will considering a large variabilities of these samples. For example, if I develop the models with solid contents with apples, so I need to involve some apple varieties with high soluble solid contents and low soluble solid content. So this is variables. Yes, so we need that to build a robust models for the future applications. So we need to consider this kind of informations. So it's not related to the numbers, because maybe if you have a large numbers, but this soluble solid is not with less variations. But if you have less numbers, but you have a large variations, it's important. This is the first. And second is to think about your application situations. For example, because there are some orchard managers ask us, that's okay, professors. So if you can help us to develop the models to manage my orchard, but actually for his orchard or her orchard, and he only grew several varieties. So we will develop the special models for him, because if we involved a lot of variations, and this will be a result, leading a result, that means you will be more stable, these models. But the stable models will not have the more precise, more accurate predictions. So if you have accurate predictions, so you will not very stable. So this is a balance. So you should find the application situations with the robustness. That's it. 2 points. So first, considering your variations or internal variations with your samples for modeling. And second is to think about your application situations. You want to develop these models for all users, or you want to develop models for some specific users. So this is not the same for modeling. Yeah, for practical. Yeah.
Okay.
Okay.
Thank you, Dr. Lan. Okay. Is there any question from the on-site participants? Okay.
Good morning, Dr. Lan. I'm Dom from the Philippines, and I really appreciate your presentation because actually I'm a first year PhD student, and
Okay.
I got a lot of, researchable areas in your presentation. And but I was just wondering because NIR or near, I just heard it a while back, and I thought it was like only for proximate analysis for non-destructive checking of a food sample, but it has a lot based on your presentation. And this is in connection with the previous question on robustness and validity, because if I remember it right, during my master's, like I'm doing this, let's say fast approach process, but they said that after this, I need to go back to the conventional method so that it could be validated. So in your case, you have mentioned that once it goes like after the technology in nears or the non-destructive technology, as long as your what did you say, it's like your data has been validated with that particular methodology, then it's good, as long as it's not about the number of samples, but it's how do you call this, the
Variations.
yes, the modelings and variation should be, you have to be accurate in your data.
If I will understand your question, and I would like to say it's not as long as means. So that means if we develop the models, and these models will be not. That means will not. That means it's not one time. It should be adjust time by times. So, for example, if you this year you build a model, but for maybe we always face the problem that if this year we build a model with rice and with their proteins, with their nutrition values, and for the next year, and this models is not works very well. That's why, because it's for the rice, it will have the different conditions, because as I said, for the agricultural conditions, were very, very complex. They will have temperatures and different rainings and so on, and seasons and different regions, so on. So in this case, and we need to collect the second years of the samples and to retraining, to put them to retraining your data, retraining your models to make it more robust. That's it. Not in terms of not only for the all users, but also for the specific users. So these both are needed. So for the years by years. When we developed the portables with the spectrometers, we cooperate with them over 10 years. So each year, we will go to orchard and to take some apples and to some fruits to characterize, to adjust, improve the models. And after that, and this will be very, very good and for the predictions, for evaluations. So that is the real practical functions of the models. Yeah, that's it.
Thank you, Dr. Lan.
Thank you.
Hello again, Dr. Weijie.
Okay.
Yeah. So, NIR, huh? I would like to ask if how technologically sound are these technologies? Say, does it affect the food quality in the long term, the health of the people who eat the food when spectroscopically analyzed, and the environment, like the fuel, the fuel needed, the energy needed to conduct these NIR? How technologically sound are these for sustainable development in the future?
You means how infrared spectroscopy will contribute to the future?
What I mean is how sustainable is it? Like, say, does it affect the quality of the food in the long term? And does it affect when someone eats the spectroscopically analyzed food?
Okay. For example, I will take an example in Chinese market. So that means if we talk about sustainable food system, one very typical or classical phenomena in China is that sometimes we buy the fruits at the supermarket, and we will have we see this fruit is good and with appearances, so we buy it. But maybe we have the different loads of the fruits. We taste it. It's not the same. Actually, sometimes it's sweet, but sometimes it's not very sweet. So in this case, so for the customers, they want to have they want to buy the products or buy the food with stable quality instead of not stable qualities. So how to solve these questions? So maybe there are lots of solutions, but one of the solution technologies is based on the non-destructive techniques. And in China, we have very good only food supermarket shops. That is called Baiguoyuan. They imported a lot of different fruits from Thailand, from Philippines as well, and so they want lots of fruits, but they use the infrared technologies to maintain their qualities. For example, they use they sell the oranges. Then this oranges means the name of this oranges is 80. It means 80 degree. What's the means? Because they use this technology for the sorting and to ensure all the oranges they sold is above the soluble salt content above 18 degrees. So that's it. That's it. Be more sustainable for the food supply chain. What we said is for the smart food processing this is my aim actually. So I think the future trends of the food processing agri-food system. That means once we harvest it with the fruit samples, we will have some good samples. We will have some not good samples. But the important is that maybe the best samples in China, we always use the selected the high qualities to sell with the higher price, with the higher price, with the best quality. And for the mediums, and we will make full use of them. And we want to doing some processings. We want doing some processings based on their features. And for the not good products, we want to sorted them. We want to sorted them and to prevent any damages or sweetens for the food safety and food safety problems. So that's it. That's we need to do that. For example, if we when we at online sorting, we have cooperation with a very large Apple online sorting companies, Orchard. And so they firstly, they will have the automatically sorting based on machine version to rapidly sorting the damage, surface the damages, and use imaging technique as well to search to find some internal damages, remove that. And then they use the infrared spectroscopy's informations to classify their content, solid content. And so to classify the grades, different grades of fruit to sell in the market with different price. And in this case, we want to say this will be control their qualities and safety and make their qualities more stable. And it will benefit for the orchard and benefit for the industry manufacturers for their higher, maybe good qualities to sell with higher prices. And yes, for them. Okay.
Thank you for your focus on food security, Dr. Weijie. And maybe we could collaborate for the environmental aspect of this technology if we could talk later.
Yes, I'm very interested because infrared spectroscopy is not only on the agricultural products, on the food. It have lots of applications as medicines, health, and also environment, waters, and some pollutions, and so on. So it's a lot of applications. So yes, because we always attend the International Near Infrared Spectrum Conference, there was lots of different colleagues from with using this kind of techniques with different areas, research areas. Yeah.
Okay. Any other question from the back?
So good morning, Doctor.
Morning.
And I'm Clyde from the Philippines. So I would just like to ask if your technology has been introduced locally, say for example, the government or industry, because I'm very interested with smart food formulation, but this question would be like general, if this has been introduced to locally, because it's like pretty challenging in the Philippines. If yes, how'd you do it? And if not, how do you plan to do it? Because it's pretty challenging in the Philippines to introduce advanced technologies.
That means you want to ask if these technologies are applied in Philippines?
Yes. No, in your country.
What is the challenges?
No, I'm asking if if you have introduced this in the government and in the industry,
Uh-huh.
and if yes, how'd you do it? And if not, how do you plan to do it?
Okay, so that means actually it's more and more popular than the previous 10 years and previous 20 years. Because at 10 years or 20 years ago, we have very cumbersome equipment, but now we can integrate some sensors in our telephone or some very micro sensors. So this is our technical background. So, and another is that now is more and more popular with data science and artificial intelligence and deep learnings. So more and more governments, producers, and also the researchers, and they notice this is important and this is valuables. This is where the data is very valuable. So now when we make some presentation or introduce the techniques to the government or to the project managers, and they are very interested because it's the new trend. It will combine the data science. It's cross-talk with it's cross-discipline with the technique, spectral technique, not only spectral technique, but also the data science, but also the chemometrics, artificial intelligence. Yes, for example, our university has for example, for the big data. So we set up the platform, near infrared. This is our latest research that we seated a platform with over 2,000 of rice varieties via infrared spectroscopy and images. There's a very large data set. And we want to combine the multi-omics, that with the spectral omics and with their qualities omics and genetics omics to have the intelligence of all data of this high output spectroscopy and qualities and the metabolism and also the genetics, and to combine all of this kind of data. So I think in the future, this is very powerful. And when we talk about this for some government and this, they will be very, very interested because it's data science. It's artificial intelligence and data science. It's a new trend because for the traditional processing techniques, they meet a lot of this kind of project. And so it's actually, it's not very attractive, in my opinion. It's not very attractive because a lot of same techniques, but for that, it's a crosstalk of different research areas. They work together. So is had that. And during this platform, we can we can find the best new varieties for the orchard. We can also control their qualities. We can also find we can combine the geneticians, the basic research and the technicals industries. So it's a bridge for them. Yeah, that's it.
Thank you so much.
Okay, is there any question? Okay, Norvin.
Hello.
Mm-hmm.
Hello, my name is Norvin, and I'm also from the Philippines. And the study you presented is very interesting. And I would just like a question. It's just I feel like this is a huge ask of a question, but is it possible if you can integrate the models with weather data or climate data to give the models more dynamic analysis of fruit quality in different environments within the post-harvest chain, for it to have like real-time analysis? Because I feel like the data you use is like in different environments, and maybe there is like a changing environment when when the fruit is stored within the producers, and when the fruit or when the produce is going to the market.
Okay. Thanks for your question. And this is very practical question, and it's from the two aspects I explained to you. And the first is that, and generally we use some sensors. For example, we use portable sensors, and generally we will have a very stable requirement conditions. For example, we will use that, and we connect it to the samples, and so we make it a very stable conditions for the light acquisitions, for the spectral acquisitions. This is stable. And in these cases, we will not always considering the environment, but for some parts, in particular with online or the moving stages or the moving situations, and for the online, we need to optimize our models. That means in some processing online or in some online sortings, then their conditions is complex actually. And so what was our solution is we have the whole equipment. We develop some online whole equipment and to make it more stable environment inside. And this is the first solution. And second solution is that we use the models and to optimize on their practical situations, and to collect the data and to retraining our data, our models. And for the mobile situation, and we want to develop, and we already developed some, but we continue to do that work, is we develop some micro sensors, and these sensors is coupled with such as processing cube champs. And so like I said, institute, so it have a dynamic processings to adjust the temperatures, grounding speed, and so it's dynamic. And so we acquire this informations time by time, and it's conscious. And so, but with also we developed some sensors such as in refrigerators, and it can acquire maybe 1 hour, maybe 30 hours time by time, and to acquire to dynamic changes of these samples. As well as we want to, now we plan to have a project with Chinese very big meat producers. That means they have the meat with in the cold track, and so we give some sensors inside of this track, and to evaluate the dynamic changes, the quality changes, freshness with the meat. So with the meat, with the temperatures and the qualities. So this is what were our solutions for that. Okay.
So for in summary, it's basically you want to like it's either keep the conditions during moving as is with how you measured it before up to the market, right? And then or create more sensors or add more sensors towards the during the moving process, so that you can account for the dynamicness of the environment. Is that it? Yeah.
That means we have we have a project with Apple producers, very big chambers, very big storage chambers with over I think 20 tons of that. So we have these sensors. And this is not our final objective. Final objective is once we acquire, we know this freshness or qualities, and we supply this, we upload this data, and we develop the models, and these models can simulate the freshness, dynamic freshness, and we adjust the oxygen and carbon oxygens, the the circumstance. So the gas conditions and temperatures and the humidities and in this chamber. So to prolong their shelf life and prolong their freshness. So this is what objective. But spectral sensors is only the detections of right now, but we combine the modelings. So it's works well right now. Thank you.
Thank you. I feel like you are at the precipice of something great, and I feel like once spectrography is spectroscopy, is that right?
Yeah.
This technology is what you call this improve more and integrated with other technologies such as like the AI and predictive technologies. I feel like it will be it will have great impacts in the agriculture industry. Thank you.
Yes, it's a very good suggestion. That means spectroscopy is generally some people will say it's just a characterized method. So, but actually we want to use this to let them to becomes more smart and becomes more artificial intelligence AI driven. So because it's the data is high output, and we want to do is that maybe we have done that. We use the hyperspectral meat, and we can monitor their whole life with during different temperatures, and we can give the predictions at different temperatures. So that is more, I think, more important than only you detect the real-time qualities. Actually, I think. Okay, thank you.
Okay, thank you. Yeah. So, due to the limited time, we have a lot of discussion with Dr. Lan. So, I'm so sorry. We will have to conclude the Q&A session here. Before we come to the end of the session, I would like to take a moment to highlight some key takeaways. So we have seen how factors such as growth condition, harvest timing, and storage significantly influence the physicochemical properties of food. However, one of the main challenges remain the limited knowledge in linking this variation to processing decision of the final product quality. So to address this, Dr. Lan has advanced analytical approaches play key role like NIR, MIR, Raman spectroscopy, hyperspectral imaging, and etc. And the key takeaways is very clear. The future food system must be data-driven, adaptive, and intelligent, where variability is not seen as a problem, but as a valuable information to optimize the process and to improve the product quality. And on behalf of the organizing committee, I would like to express our sincere gratitude to Dr. Lan for his valuable and inspiring presentation. And we hope today's discussion inspires further collaboration and innovation. And let's give a round of applause to our speaker.
Thank you. Thanks for all.
Okay, thank you very much. And I would now like to hand over the session back to the MC.
All right, thank you so much to Mr. Weijie for being insightful lecturer about from Spectra to decision, AI-driven chemometrics for fast classification, prediction, and traceability. I would like to give the appreciation certificate that will be given by our moderator. Maybe give applause for the speakers. And also, I would like to thank Dr. Lulum Leliana, who led the discussion before, and the certificate of appreciation will be given by the committee of the short course 2026, Ms. Hilda Maya Cynthia Dewi MSc. May we give applause for the moderator today? With the session of appreciation certificate already implemented, then the first material of this summer course has ended. For operators, you may stop the recording